Hello, this is Dr. Malescu, and I'm just going to do a little micro-learning lesson on the cell cycle. And we're going to begin with explaining G1 as G2, and the small phase of mitosis gets a lot of attention. So I'm going to start with first the other aspects of the cell cycle that is not so dynamic, where the cell is kind of prepping the entire time to get ready for cell division and duplicate itself, literally creating two equal daughter cells, genetically identical, with the 46 chromosomes from the original cell. So G1 is the first phase, S is the second phase, and G2 is the third phase. These phases right here, it's called the preparatory phase, and it's also known as interphase. So interphase equals G1, S, and G2. The way I remember, S is sex. So this is when the DNA is actually replicating itself. When we replicate ourselves through sex, we create offspring. Well, the DNA just duplicates itself, basically, not to be confused with protein synthesis, where the messenger RNA is transcribing the DNA and all that. No, we're not talking about transcription and translation protein synthesis. We're talking about the DNA that is a double helix, like so. So I'm going to do two different colors. So here's the DNA double helix, okay? So when you have DNA synthesis, we're now going to create one strand here that has unraveled, and then the other strand in red that has unraveled. And now we have two new strands building up on the single strand. So we have now two double-stranded DNAs that are pretty much genetically identical. So for this one, we're going to build a new DNA. So now we have a new DNA double strand that's ready to go. And then for this single strand that unraveled from the original, blue, is now going to get its own base pairing. And so we base pair the whole process. And now we have two DNA double helixes that are ready to go into that mitotic phase, where when the cell finally divides, we have the genetic material that's identical in both cells. All the DNA is a double helix. So that's the S phase, synthesis, DNA synthesis, okay? Now I'm going to withhold talking about G1 and G2 just so I get to the next slide, because this is where the cell is prepping. So we're going to have like organelles duplicating themselves, increased cellular metabolism, more protein synthesis, and things of that sort going on in G1 and G2. So I really want to focus heavily on the DNA synthesis aspect so that you understand that process, because that is the crux of the ability for the cell to actually go through division. It cannot go through division unless the DNA is prepped. So here is a chromosome, which is basically DNA all coiled up in this very large chromosome. And the center is pretty much called the centromere, not to be confused with centrosomes that are located in a cell that form the spindle and allow the chromosomes to move during mitosis. And so these are homologous chromosomes, okay, which means that they are base paired and they are identical in terms of information. It's just that they're bound by their sister chromatid, which was produced during that DNA synthesis or replication during the S phase. So in the end, G1 is the process where the cell is not actively dividing, but it is performing cell growth. Okay, so if you want to dive deep into that, you can. And G2 is there's more protein synthesis, so an organelle duplication. So we have more growth in terms of more mitochondria buildup, so that the cell has energy for mitosis phase. So the mitotic phase is PMAT, a puppy pees on a PMAT. So that mitotic phase is right here. It's a small little blip in time And I like to say a puppy pees on the PMAT because it's easy to remember. PMAT stands for prophase, metaphase, anaphase, telophase, PMAT, okay? So the highlights of prophase is that you're going to start to see the chromatin coil up into visible chromosome. The nuclear envelope around the nucleus disappears. And in the late stages of prophase, you're going to start to see the centrosomes going to the opposite ends, and that's going to be a preface for the spindles to form, so that in late prophase, you're going to start to see the spindles. Metaphase is right in here, and metaphase is the cell, the chromosomes within that cell come to the equator, to the center. So chromosomes move to center, okay, meaning the equator. All right, so prophase, we had nuclear envelope breakdown. We had chromatin condensing to chromosome. And then late stages, don't forget, which I'm not going to write down, is that, you know, you're going to start to see the centrosomes right here. Centrosomes move towards opposite polar ends. And so that's going to allow the spindles to form. And once the spindles form, they pull the chromosomes to the center, and that's metaphase, move to the center. Anaphase is Anna gets a divorce. That's how I remember. So the chromosomes, the sister chromatids separate from the centromere, and the chromosomes go to opposite polar ends of the cell, right here and right here. And then in telophase, you have the cell pinching in. We call that cleavage furrow, because it kind of looks like cleavage of two breasts. And then that cleavage furrow forms to cause the cell to pinch in. The nuclear envelope reappears, so it's the opposite of prophase. The chromosomes become chromatin. They start to look like linguini again. As the nuclear envelope reappears, the nucleolus reappears, the chromatin is now visible instead of chromosome. The spindle fibers are going to disappear. And then final phase is cytokinesis. Once the cell is completely pinched in, the cell separates, and you've got 46 chromosomes in one cell, 46 chromosomes in the other cell. And basically, you've got an even number of chromosomes with two equal daughter cells that are genetically identical. And that there is the cell cycle. I don't think there's much else to say other than giving you a few more slides just to solidify and take this home for you so you can understand this little micro lesson on cell cycle. So the checkpoint is the decision whether to continue the cell cycle. So I want to mention apoptosis. Apoptosis is a term, cell suicide. So if there is unregulated and continuous cell cycle forever, the cell begins to think it's immortal. That's what cancer is. Cells gone wild is cancer, and it is cell cycle continuously going, and there's no cell suicide. Okay, so know that term, cell suicide, apoptosis. Okay, what else did I want to mention? Like I said, there's cell growth in G1 and cell growth in G2. If you want to do a deep dive as to in what location the organelles are growing, and in other parts of the G1 and G2, you have increased cell metabolism. All right, so we want to increase, right? It makes sense. We want to increase energy for that big event, which is mitosis. Okay, so moving on, you're going to see other types of cell differentiation that comes from stem cells, which is a bit different than the cell cycle and mitosis. So just so you know, the red blood cells, the white blood cells, and the platelets all differentiate from one stem cell in the bone marrow. And so that's very different. So just make sure you remember that the process of hematopoiesis involves the development of different types of blood cells arising out of the hematopoietic stem cells, very different than the cell cycle. Okay, and that's pretty much all I want to present today because I really want to keep it short and simple. All right, so don't forget cell cycle, G1, S, G2. G1 was cell growth, S is the DNA synthesis or replication, G2 is the second cell growth portion of the cell cycle. The gray area here, G1, S, G2, is the entire part of the cell cycle is called interphase. And then the mitotic phase is PMAT, prophase, metaphase, anaphase, telophase, and cytokinesis. Now you could add this other pro-metaphase, but I like to keep it simple, keep it KISS, keep it simple, stupid. The chromosomes condense, become visible. They turn from chromatin into visible chromosomes. The spindle fibers begin to form because these centrosomes are moving to polar opposite poles. The nuclear envelope breaks down, the nucleolus disappears. Pro-metaphase, the chromosomes continue to condense, and the spindle is now quite visible. And then in metaphase, the chromosomes go to the equator, to the center. Anna gets the divorce. The chromosomes separate. The sister chromatids go to opposite polar ends of the cell. They separate at the centromere. And then after anaphase, we have the cleavage furrow, where the cell starts or begins to pinch in. And now what's going to happen is there's going to be a nuclear envelope forming around the sister chromatids. Okay, and once the nuclear envelope reforms, the cleavage furrow forms, the nucleolus begins to reform, and the chromosomes turn back into chromatin. So the nuclear envelope material surrounds each set of chromosomes. The mitotic spindle breaks down, and therefore the next final step would make sense, the cell separation. Cyto meaning cell, kinesis is movement. So movement of the cell to divide into basically the two equal genetically identical daughter cells. All right, so that's cell cycle. I hope you enjoyed this little micro lesson. Bye.